Hello everyone, and welcome to our group's presentation. Today we're going to be presenting about the review article titled Enzymes in Food Processing, a condensed overview on strategies for a better biocatalyst. Now that's a lot to take in, that's a pretty hefty title, but please remember to keep in mind the previous presentation, which is also about enzymes. We're going to be talking about enzymes as well, and we're going to be following a similar train of thought here. And believe it or not, we are surrounded by examples of enzymes involved in food processing every day, more than you might even know. And you know a lot more about this than you think. There's a lot of foods we consume that are created by enzymes involved in the food processing, from bread to coffee to tea to candy to a lot of different foods. We have enzymes to thank for a lot of different foods we consume, and it's really fascinating and really interesting, and we're going to delve into a little bit more of that today. This article specifically focuses on the idea of application of biological agents becoming more effective and diversified over time. Specifically, this article is aiming to look at how enzymes, specifically biocatalysts, and their applications and how they can become more efficient and more effective over time. In other words, how can we take something we have now and make it better? Some of the enzymes we might be looking at in this article you might already be familiar with, like amylase, pepsin, and xylose, just as a few examples. We'll get into more hefty details as this presentation goes on. The next slide is going to focus a bit more on biocatalysts, a word I've mentioned already, but we'll go into with some more depth in the upcoming slides. So be prepared, take some notes, and please enjoy.
Hi everyone, my name is Hannah Brantley and I'm going to be discussing the relevant enzymes involved in improving biocatalyst and food processing. So first, just some general information. Almost all enzymes play a role in food processing. Hydrolases are usually the most favored, and this is because they act on carbohydrates, lipids, proteins, and nucleic acids, and one of hydrolases' main functions is digestive, to break down these nutrients into smaller parts for digestion. The use of enzymes in food processing is pretty understandable given their specificity, ability to operate under mild conditions of pH, temperature, and pressure while showing high activity and turnover numbers, as well as high biodegradability. Enzymes are also considered a natural product which contributes to their sustainable and environmentally friendly processes, since there is a low amount of byproducts and the energy requirements are relatively low for these reactions. The life cycle assessment confirms that enzyme-based technology has a positive impact on the environment, and the majority of enzymes used in food processing is of terrestrial microbial origin. Next, I'm going to talk a little bit about the importance of screened enzymes. These enzymes are important because they operate at high temperatures, roughly above 45 to 50 degrees Celsius, which minimize the risk of microbial contamination. These reactions are relevant to food processing because they favor the isomerization of glucose to fructose. Some examples of screened enzymes include the isolation of amylases, with some of them being calcium independent, amylopulinases, fructosyl transferases, glucoamylases, xylose isomerases, glucosidases, leaven sucrases, and xylanases. Next, I'm going to be discussing some of the specific enzymes and their functions used in food processing. There are 5 different classes of these enzymes, oxidoreductases, transferases, hydrolases, lyases, and isomerases. These enzymes have many functions, but they are widely used in several food preparations for improving taste and texture, and they also offer huge economic benefits to industries. Enzymes that are oxidoreductases include glucose oxidase, lactases, and lipoxygenase, and glucooxidase plays a role in dough strengthening. Lactases play a role in as a flavor enhancer, and lipoxygenase also plays a role in dough strengthening and bread whitening. Transferase enzymes include cyclodextrin, glycosyltransferase, fructosyltransferase, and transglutaminase, and these function as synthesis of fructose oligomers, dough processing, and meat processing. Hydrolases include amylases, galactosidase, glucanase, glucoamylase, invertase, lactase, lipase, proteases, pectinase, peptidase, phospholipase, phytases, polulinase, and xylanases. Amylases play a role in starch liquefaction, increasing shelf life, and improving quality by retaining moist, elastic, and soft nature of the food. It functions in the softness of bread and bread volume, flour adjustment, ensuring uniform yeast fermentation. Galactosidase is important for viscosity reduction in lupins and grain legumes used in animal feed, and also enhances digestibility. Glucanase is important for viscosity reduction in barley and oats used in animal feed, and also enhances digestibility. Invertase is an enzyme that functions in sucrose hydrolysis and production of invert sugar syrup. Lactase functions in lactose hydrolysis and whey hydrolysis. Lipase gives cheese flavor, and proteases are important for protein hydrolysis, milk clotting, low allergenic infant food formulation, enhanced digestibility, flavor improvement in milk and cheese, meat tenderizer, prevention of chill haze formation and brewing. Pectinase is important for juice clarification. Peptidase is important for the hydrolysis of proteins, mainly soy and gluten, for savory flavors and cheese ripening. Phospholipase is important for the emulsification for dough conditioning. Phytases release phosphate from phytate, which also helps enhance digestibility. And xylanases are important for viscosity reduction, enhanced digestibility, and dough conditioning, as well as some of the others. Lyases are important for beer maturation, and that is by the enzyme acetyloacetate decarboxylase. And then xylose isomerase is one of the isomerases, and that is important for glucose isomerization to fructose. So as you can see, most of these enzymes are very similar in their functions for improving taste and texture of food.
Caven Price, and I will be discussing improvements of biocatalysts. Biocatalysts have been consistently improved by utilizing the information regarding molecular biology, high-throughput processing, computer-assisted design of proteins, and in vitro. So there are three main factors that scientists look at for when improving biocatalysts. They have stability, range of pH, temperature of operation, and enzyme activity. So when you talk about stability, the focus is mostly on thermal stability. And thermal stability is the quality of a substance to resist irreversible change in its chemical or physical structure at a relatively high temperature. Enhancing the stability of an enzyme is important because it reduces the amount of enzymes used in the process. Biocatalyst stability is enhanced by protein engineering. Protein engineering is used because it can enhance enzyme stability without compromising catalytic activity. Another property researchers improve for biocatalysts is broadening the range of pH where the enzyme is active. The last factor that we're going to look at is the temperature of operation and enzyme activity. Scientists want to decrease the temperature of the operation while keeping the enzyme activity high. And they want to decrease the temperature of the operation because at low temperatures, the risk of contamination, microbial contamination, decreases, and the rate at which raw materials lost is decreased as well. So there are two methods of protein engineering, and they are random mutagenesis and recombination and rational pinpoint modifications. So during random mutagenesis and recombination, environmental adaptation is reproduced in vitro quickly with a focus on optimization of the intended property. So also screening tests and selective pressures are used for random mutagenesis and recombination, and it is aimed for more efficient biocatalysts. On the other hand, rational pinpoint modifications are alterations promoted and performed based on the growing knowledge on the structure and function of the enzymes. So, another important part about rational pinpoint modifications is that it utilizes computers. Like, it is basically computer science, computer research. And even though computational research is fairly new, it is credible. And so, this research method is especially helpful, and it helps better lead random mutagenesis research. It is also important to note that these two methods are not mutually exclusive. Methods for engineering can be used to assemble both strategies, and what this basically means is that you can combine these two strategies to come up with the improvement that you want. So you might use both rational pinpoint modifications and random mutagenesis and recombination to improve a biocatalyst. This chart just further sums up what I was trying to say in the previous slides. It is basically this chart basically shows you the enzymes, the role the enzyme plays, the target improvement that the scientists want, and they showed the strategy that the scientists use to get the target improvement. An example that we can look at really quickly is phytase. It's the enzyme. Phytase plays a role in animal feed, and the scientists wanted to change the pH activity of phytase, and they did this by using protein engineering through site-directed mutagenesis.
My name is Edith, and I will be talking about enzyme immobilization. The process of enzyme immobilization involves improving the stability of an enzyme, but is also a way to reuse the enzyme in a bioreactor system. An immobilized enzyme is an enzyme that is attached to something insoluble and unmoving. There are a few benefits and a few drawbacks that were discussed in the article relating to immobilization. A few of the benefits include that the enzyme can be easily reused. There is also easier separation of the enzyme, and immobilization helps to resist denaturization of the enzyme. Some of the drawbacks that were listed include that the enzyme can lose activity during immobilization, and that depending on the method of immobilization used, it can be quite expensive. The three main methods of immobilization that were discussed in the article include entrapment, carrier binding, and cross-linking. Entrapment is when the enzymes are held in a substance, gel, or capsule. Carrier binding involves the formation of covalent bonds between the chemical groups in the enzyme and the chemical groups on the carrier. And then cross-linking is the covalent bonding between molecules, often using glutaraldehyde. And this cross-linking method is the method that is used most often because it is the least complex of all the three. So bioreactors are the place where the reaction takes place, and bioreactors are often used in food processing. There were two main types of bioreactors that were discussed in the article: the batch operation and then the continuous operation. The batch operation is when the bioreactor is filled with an enzyme and then sealed, and then the reaction takes place in the sealed environment. When the reaction is finished, the bioreactor is opened and then the product is removed. In the continuous operation, the substance will flow in at one end of the bioreactor. It will be processed and then will flow out of the other end of the reactor. This continuous operation is more efficient, however, it takes more hands-on time. This figure shows different kinds of bioreactors. Figure A shows the packed bed reactor. This is when the enzyme is bound to beads. The substance will then flow through the reactor and then through the beads. Figure B is quite similar to Figure A, however, the beads can move freely in the bioreactor. Figures D and E depict the batch operation, where the bioreactor is filled with an enzyme and then sealed, and then the reaction will take place in this enclosed environment. Since there is not a continuous flow of fluid in the bioreactor, when the reaction is finished, the enzyme will have to be discarded.
Alrighty, so moving along to the conclusion of our presentation, enzymes and food processing. We did go over a few things, so the relevant enzymes, classes of enzymes, improving biocatalysis and immobilization. And so this conclusion just goes over briefly some of those highlights and key points of those sections that we discussed earlier. So findings are particularly important because it allows enhanced performance under operational conditions, which reduces the risk for bacterial contamination. We also discuss how it favors process integration by allowing the concerted use of enzymes that naturally have diverse requirements through development in molecular biology, enzyme engineering, and computational tools. And then next, we also discuss the mobilization of enzymes through those three methods: entrapment, carrier binding, cross-linking of enzyme aggregates, as well as bioreactor, which was broken up into the continuous mode and batch operation. And so with this, immobilization of enzymes is a supporting tool for rendering these proteins while supporting improvement for their catalytic functions or features, excuse me. And we also went over the 5 classes of enzymes used in the food processing, which includes oxidoreductase, transferases, hydrolases, lyases, and isomerases. And then also we have to consider that that despite the developments made in the field, there's still a lack of applicable rules for enzyme immobilization, which we discussed, which includes technology and economic requirements. But when we think of the economic requirements, it's more so has to deal with food in the food sector. And then the last bullet point, the relevant enzymes and its use in food processing. It can be foreseen that the efforts will be towards the developmental of immobilized biocatalysts with suitable chemical, physical, and geometric characteristics, which can be produced under mild conditions that are used for different reactor configurations and that comply with economic
requirements for large-scale applications. So that concludes our presentation on enzymes and food processing. And this is just a site of our references that we used, of course, the article. Thank you.